This is a production of Cornell University.
Okay, thank you so much, Teresa. It's quite an introduction. I am confident that I'll live up to it in this presentation about pockets because it's one of my favorite things to talk about. So, although Teresa's right, I haven't worked in this system for a little bit. Most recently, I've been working with a different kind of symbiosis with endobacteria and arbuscular mycorrhizal fungi. You'll hear more about that next time. The timing is fortuitous here because I've got a chapter coming out soon, a book chapter on these little organs, these pockets. You can see them here on the silhouettes. Here, the circle starting again. Oh, never mind. I also started myself. Well, pockets. I think that these fungus pockets are completely underappreciated in ambrosia beetles. And I think ambrosia beetles are far too obscure in the first place. So what am I here to do? I'm here to tell you about fungus pockets and sell you on the fact that they are the greatest symbiotic organ that's ever existed and that everyone should know about them and tell other people how cool they are. And so let's see how I do on that. Here's a photo of a beetle. It's a cute little beetle, right? Notice there's something in the front of the beetle, the pronotum area. It almost looks fake. It looks like a paper clip inside the beetle's body or something. What is that? Hey, here's a 3D image of whatever that thing is. That looks wild. It's like a hose pipe rolled up inside of the body. Here's a photo through a microscope. You can see it through the transparent exoskeleton of the beetle. What the heck is that? Here, the tube's been taken out and it's been smashed under a microscope slide, and what look like fungus spores are being squeezed out like toothpaste. What is this mysterious tube? Well, I'll get back to that. Here's a beautiful illustration showing some wonderful wood insects. Notice the beautiful design some of them make on wood, these engraving beetles, as they as their larvae dig tunnels through the phloem in the inner and outer bark. Notice the same up here, beautiful patterns in the bark, beautiful patterns in the bark. These are bark beetles. Maybe you've seen their evidence around town when you've peeled a piece of bark off the tree. And if you haven't, start peeling in places where you're allowed to peel bark and you'll eventually find some works of art. But something different is happening at the bottom right. These tunnels are going straight into the tree. And that's weird because there's not a lot of food there. Phloem's great. It's full of sugary phloem. And that's a good food source. It makes sense that there'd be a lot of competition to want to live there, but who in their right mind would dive into the sapwood, the dead part of the tree where there's almost nothing? Who indeed? Well, I can tell you the answer is ambrosia beetles. And ambrosia beetles don't just live there. They continue to live there and they pack up all the time and live there again. And it's not by living on the terrible nutrients inside of that wood. It is by farming fungi, actual insect agriculture. The fungi concentrate the nutrients from the wood into something the beetles can eat. The beetles eat the fungi. It's a simple relationship. We do the same thing with corn. We don't eat soil generally, right? But corn kind of does, and we like to eat corn. It's the same relationship. The beetles are farming a cultivated fungus to extract nutrients from this sapwood. Now, this ambrosia fungus, it's very delicate and it can't disperse on its own. It's become so good at becoming delicious food. It makes these huge, juicy spores that the beetles love to eat. But these spores are so big, they just collapse and die the instant they hit dry, open air. They need to be in these humid tunnels to survive. And considering there's so many other hardier fungi waiting in the wood to take over, to cover the tunnels, to attach themselves to the beetles, how the heck do ambrosia beetles fly to new trees almost every year and start new, perfectly pure gardens of the same fungus cultivar? How do they keep their crops? I'll get back to that. Let me tell you about Laurel Wilt, because I know this is a plant pathology seminar, and I've made sure to include some plant pathology material. Despite being part of plant pathology departments for, some years, I'm only kind of a plant pathologist. That's what happens to mycologists sometimes, right? but almost anyone should have heard of Laurel Wilt in the plant pathology world. This is an Ambrosia beetle fungus combo introduced to Georgia in the early 2000s that within 15 years killed 300 million red bay trees and threatens every other member of the Lauraceae. That includes things like camphor, sassafras, and, oh yeah, avocado, a tree that a lot of people have a good relationship with. It was thought to have originated with a single introduction from a shipping container, maybe even a single female beetle. How is that possible? How could a single beetle start an epidemic? I'll get back to that right now, because the answer to all three of these questions that I posed so tentatively is the same answer. What is this mysterious tube? How do ambrosia beetles keep their crops? How can one beetle start an epidemic? Pockets. Did you expect anything else? It's the topic of this talk. Just like we use pockets of different sizes to carry different things, ambrosia beetles use pockets that are kind of like the artist's depiction here to carry fungi. And just like different size pockets carry different things, different ambrosia beetle fungus pockets carry different fungi. And that's really important. And it is well worth learning about and understanding what these pockets are and how they work. So, powerful pockets. How these pockets define fungus farming in ambrosia beetles. Let's start with a pretty important question. What are ambrosia beetles anyway? I've been using that word a lot, like everyone knows and is familiar with it. Well, it's simple. They're beetles that farm fungi in xylem. Kind of, because there's a few caveats. They tunnel into sapwood. Sometimes they will go into heartwood or pith, and they farm lush gardens of ambrosia fungi. Look at the beautiful white lawn here. They do have parental care and various levels of sociality. There's even a eusocial species that has an ant colony-like multi-generational lifestyle, but it's just one species. But here, the mother beetle is tending to these larvae, which are enjoying their entire life in one little cradle with all the food they can ever need growing around them, the ambrosia. Here you can see how they tunnel deep into the wood itself and the bright white fungus growing inside. Are ambrosia beetles a taxonomic group? That would be convenient. Absolutely not. Ambrosia beetles evolved more than 12 different times in two families of true weevils. That's right, they are true weevils that have simply lost their snouts because snouts kind of get in the way when you're digging in wood. The first is the family Platypodinae, and this unrooted tree here from Holzer and Stilinski 2017. You're going to see more of this tree. Here's the Platypodinae all by itself. All of them are ambrosia beetles, every single species. But starting here, and everything else in this tree is the family Skeletinae. And of those 6,000 Skeletinae, about 3,500 are ambrosia beetles. The rest are mostly bark beetles. There's a few that are things like seed borers and pith borers and things, but most of the rest are bark beetles. Now, you might be wondering, if you're somewhat familiar with the system, well, aren't ambrosia beetles bark beetles? Kind of. Bark beetles is used colloquially to refer to the entire Skeletinae, which includes all of the ambrosia beetles within. But ambrosia beetles aren't really in the bark, and the original definition of ambrosia beetle was to distinguish it from bark beetles. So I don't consider ambrosia beetles bark beetles. Instead, ambrosia beetle is an ecological term that for most of its history has been used to distinguish them from something else, bark beetles. So what's the difference? The problem with using a definition based on being not something else is that it gets pretty messy over time, and I'll explain in a bit. You certainly can't just look at a skeletine and decide if it's an ambrosia beetle or a bark beetle, unless you're an expert, of course. They look really similar. These are examples of both that we can find here. And to most people, they just kind of look like weird beetles. I mean, this one looks like it's gone through an accident. It's kind of been amputated. It uses that surface to plug the entrance of its tunnels in from intruders. So it's basically like a built-in door, but they're visually similar. The evidence they do in nature because of their different ecology is not similar. Here's a photo I took in my yard here just like a couple months ago after peeling some bark away. Those are the beautiful engraved galleries of bark beetles. Here we see what are commonly known as frass noodles or toothpicks. Ambrosia beetles dig really far into the wood and they have to push all that frass out. And for whatever reason, that frass sticks together really tightly and it forms little noodles. So if you ever see noodles sticking out of a tree and you think someone's made some kind of prank with spaghetti, it is almost certainly ambrosia beetles. I took this photo in near Cliffs of Varna up in Monkey Run here in Ithaca. But this difference has been appreciated for a really long time. This photo is, actually, I don't know the source, but it's public domain. But beautiful vintage art here showing the difference between bark beetle and ambrosia beetle galleries. And now might be a good time for me to demonstrate, depending on if you have the speaker camera on. If you do, I have the whole right side of the slide available for you to put the video bar in. But here's a nice example of some ambrosia beetle tunnels. Well, in this case, they're actually cradles where the larvae would eat. They're extremely small. Here's a bookmark that I got from a good friend and collaborator, and you can see there's a cross section of a few ambrosia beetle tunnels there. They're really, really little. And they're ecologically very different from bark beetles. So I'm going to return to this beautiful picture again because it illustrates this difference, all of the bark beetles versus the ambrosia beetles in the wood. This was recognized more than 100 years ago. This is Hubbard in 1895 in the paper that defined the word ambrosia beetle. The ambrosia beetles of the United States. And Hubbard simply defines them as a term to distinguish them from the true bark borers, as those that push their galleries deeply into the wood and feed upon ambrosia, which was known to be a fungus. So that's the two definitions, right? Easy. So all ambrosia beetles tunnel into the xylem. All bark beetles tunnel into the phloem. That's great. All ambrosia beetles are obligate fungus farmers. So it only makes sense that all bark beetles are not obligate fungus farmers, right? Well, this is where it starts to get messy because some bark beetles benefit or require fungus to develop fully. A lot of them have really tight associations with fungi. They get just about as close as you can get to being called farming. They even have, well, I almost got ahead of myself. The symbioses with fungi with bark beetles can be extremely complex. And so really the only thing different is they're doing it in the bark. It starts to become a little arbitrary. All ambrosia beetles have fungus pockets. Surely this must be something unique to ambrosia beetles. No, some bark beetles also have fungus pockets, some of them extremely complicated and complex, especially those that rely on those fungi. I say all this because these words are mixed up all the time in the ambrosia beetle, bark beetle world, and it's kind of understandable because the line between them is very blurry. And it's based entirely on a group of more than 12 different unrelated lineages that do the same thing to put them apart from something else. But keep all that in mind because I'll be mentioning ambrosia beetle and bark beetle. That's right. Sorry, I got totally off topic. I'm supposed to be talking about pockets. So, okay, so pockets full of pathogens. So do all disease-causing bark or ambrosia beetles have pockets full of pathogens? This is a common assumption. After all, maybe the most famous of bark and ambrosia beetles cause disease. And if they have symbioses with fungi, then it must be the fungi causing them, right? This is really not generally the case. Let me first introduce Dutch elm disease, Ophiostoma novo-ulmi. This should be very familiar to everyone, including important work that was done here on the early disease. Pictured as just one of the 3 vectors in the US. This does not have pockets full of pathogens because it doesn't have pockets. This beetle does not have does not have mutualism with the Dutch elm disease fungus, and it doesn't have the fungus pockets to carry it. Dutch elm disease is just really good at sticking to the surface of beetles. That's part of why it can be vectored by 3 different ones. It has really sticky spores. The fungus certainly benefits from the transmission, but it's unclear if the beetle's getting much in return, especially in the way of nutrition. Laurel Wilt, yes, it does have pockets full of pathogens. The same fungus that this red bay ambrosia beetle introduces into trees that kills the trees is the fungus it farms and eats to survive, and does in its native range as well. So yeah, the fungus that causes disease and the mutualists are one and the same. Southern pine beetle, that's one of the many destructive bark beetles in the US. Pockets but no pathogen. It has incredibly complex pockets. They look just as amazing as some of the most large and ambrosia beetles, and it definitely holds two different mutualists, one an ascomycete, one a basidiomycete, that the beetle feeds on. But it's not the fungi that cause the majority, if any, of the disease. It's the mass attack by the beetles, the physical damage. And finally, the black stem borer, which should be very familiar to some here, because there is at least one, if not more, groups here at Cornell and Geneva working on black stem borer, especially where it's a nursery or orchard pest. It does have pockets, and it does have a mutualist that it farms to eat, but that mutualist is not the fungus that causes disease. In fact, again, it's the beetle itself that causes most of the mechanical damage that actually dooms trees, although it gets even more complicated because it can vector some somewhat pathogenic fusarium, either on the surface or in the mycanjum along with the mutualist. So now you can start to see why this entire thing is full of caveats everywhere, and I can only cover some of them. But I think it's important because you know how we learn more about all these things? It's by learning about the pockets. I'll get back to that. Of course, again, plant pathology seminar, I want to touch on the economically important ambrosia beetles in the USA. Pests of timber, many species, too many to talk about. Ortholis columbianus is a well-known one because it makes little flags in the wood. It's called flagworm often, and it'll attack living trees sometimes. Most of them are attacking timber, already cut, felled, or cut trees. Nursery and orchard pests, there's two big ones in the US, the granulate ambrosia beetle and the black stem borer that I mentioned before. Both of them are really serious pests in nurseries and orchards especially. There's a few others, including Anisandrus mache, which only just arrived here, hasn't been too much of a problem yet. Nessus mutilatus and Xylosandrus compactus are other invasive pests. But these two are by far the biggest problems. But these are the signal alert pathogens right now. These are the big deals in the country, and they're on both sides. They're on both shores. The first, Laurel Wilt, which I mentioned. Red bay ambrosia beetle vectors the Laurel Wilt pathogen Raphaelia luricula. The second are two different polyphagous shothole borer species in California. Well, I should say the polyphagous shothole borer and the Carisio shothole borer. I'm just going to use PSHB for now on for this because that's a lot to say for both. They both vector vascular fusarium dieback pathogens, and they're causing big problems in California. Note, I'm not going to list all the bark beetles that cause big disease. They are bark beetles. That includes thousand cankers disease, mountain pine beetle, southern pine beetle, etc. We're just going to focus on ambrosia beetles. The thing about pests of timber is that this has been known for a long time, since the early 1900s. And it's been known that they're really picky about that wood, that the wood has to be of excellent quality and recently killed and just moist enough, and it hasn't dried out too much. And then the ambrosia beetles will attack, dig in, make their galleries, and ruin that wood. I mean, although I like it. I like the wood for making crafts with. Generally, it would ruin timber, and it causes millions of dollars of damage. But this is the normal biology of ambrosia beetles. They, with only a few exceptions, are geared to attack stressed, dying, and recently dead trees. That way they don't have to deal with tree defenses. They're going for trees that are already on their way out or are stressed and can't defend themselves. For this reason, ambrosia beetles are really frequently blamed for killing trees when they're almost always innocent. But you see a tree die and suddenly it's got a bunch of noodles coming out of it, spaghetti noodles. It's easy to blame the beetles. Mass attacks, though, this is another problem. So both of the major nursery and orchard pests do this. Look how many noodles are coming off of that tree. There's no way that can be good. So this basically girdles the tree by punching so many holes in it, making so many tunnels that the vasculature can no longer support the tree. They attack in huge masses and just overwhelm the tree. The same thing happens with the polyphagous shothole borer, but the effects of the fungus are a lot more evident. But still, the fungus is not a one inoculation kill. It works in tandem with mass attacks by the beetle to overwhelm the tree's defenses, cause these terrible scars and dieback, and eventually death. Now, in each case, fungus can help this mortality. Maybe the non-mutualist fusarium in the granulate and the black shot-hole borer, and the black Ambrosia beetle. That's not super well shown, but sure, they have fusarium associated with them and it can cause symptoms. It's certainly not the mutualist fungus. Polyphagous shot-hole borer, definitely the fungus aids in the symptoms, and it is the nutritional symbiont that they farm as well. But finally, the true wilt disease, Laurel wilt, the only true wilt disease vectored by an Ambrosia beetle. There is another Raffaellia disease in Japan, Raffaellia quercifera, but I'm going to stick with the US for now. So this is a true wilt disease. The fungus is the killer. Now, whether it's because the tree commits suicide by making too many tyloses in response to the fungus, or the fungus gums up the vasculature, well, I'm not the person to ask. I'm not sure if it's still in the literature. Remember, I'm only kind of a plant pathologist, but this is definitely a big deal. It runs rampant on Lauraceae trees. Look how brown those leaves are. Look at that open part of the tree with all the tunnels. This is a big threat. I asked earlier, how can one beetle start an epidemic? Well, now I can actually answer it fully. It's not just pockets. Because one part is that the red bay ambrosia beetle, like all xylobrini ambrosia beetles, the females can start, or they're haplodiploid, so they can start an entire population on their own if they're pregnant. Add Mycanja in with that, carrying the perfect fungus from the native country, and when the beetle invaded from Asia, it already had the pathogenic fungus, the fungus which trees were pretty resistant to in Asia, ready to go. So the answer is pockets. So I want to quickly cover the publications per year that include the word ambrosia beetle, because I think this tells an important story. The word was defined in the late 1800s. Yes, here in 32, black stem borer was introduced to the US here in New York. Okay, 1932, that's supposed to be 1974. That's okay. The granulated ambrosia beetle was introduced in South Carolina. Both of those nursery and orchard pests. Suddenly, Laurel wilt hit. It was noticed a few years earlier, but this is the year it was really appreciated. And suddenly we have something. Then 2012, polyphagous shot-hole borer in California. And it's been nothing but up since then. Now I did my PhD in this region, started about right here, just in the most of the increase. Now I don't want to leave out, because again, this is just a search on the number of publications. Obviously, worldwide disease could have an impact on this too. So their Japanese oak disease, the other Raffaelli disease, did happen in the late 90s. Polyphagous shot-hole borer has hit Israel and South Africa. And of course, those have contributed to interest. But what I'm trying to say here is that Laurel wilt acted as a spark that incited a lot of interest in this system that hadn't been there before, despite the orchard and nursery pests, despite the amazing symbiotic model system, which I'll get to in a bit. It wasn't until these really big diseases hit that suddenly ambrosia beetles really increased in interest. And I checked, this is more than the already exponential increase in publications in the world in general. So it's an ambrosial renaissance of some sort. But for some perspective here, there's 5 serious pests in the US. All of them are invasive. I talked about all 5. There's about 30 invasive ambrosia beetles total in the US and Canada. There's 86 total ambrosia beetle species, including natives. And there's about 3,500 ambrosia beetle species worldwide. And the interest that's been put into research in understanding basic biology is very outsized for these problematic species compared to the rest. And they often miss the significance of pockets in the ecology of these beetles. Oh, right, pockets. Okay, I got way too far into plant pathology. It's time to get back to pockets. So what's in a pocket anyway? It's about time we talked about that. I keep calling them fungus pockets, and maybe you're really envisioning like pants pockets or something, but no, they're a lot more amazing than that. Let me cut this beetle open here. And we see a strange little structure. If we put that on a microscope slide, if we squish it down and look closer, it is a membrane filled with pure fungus spores. This is the mutualist. This is the ambrosia fungus that the beetle farms. But that is far from the only example of a fungus pocket. Here we have two little pockets just at the top of the wing covers. Here we have a little bowl on the side of the body covered in hairs. Here we have just two little perfectly circular dimples that hold spores. You remember the fungus tube from earlier? There might be a clue as to what that tube is all about. Here is a couple more shots of the one I just showed you. It's a big membrane inside the body. Here we see membranes near the mouth, so while the beetle's eating, some spores can go through the esophagus and get stored. Winding tubes, big hairy patches. There's a lot of different kinds of fungus pockets. Here's a color-coded list of the most well-studied. And I know it's a lot, but just appreciate the diversity here. Winding hose pipes, horseshoes, little fins, something that looks like a UFO upside down in the beetle. It's incredible how different these are. A little sphere inside of the head. All of them separately evolved to hold fungus spores. That's why they look so different, and that's why they're so incredible. How did they get discovered, though? Well, it was an Austrian monk in the early 1800s who noticed the beetles eating this white stuff that he called ambrosia. It was the mycologist Hardig who said, well, that's fungus. It's not sap, it's a fungus, and it generates spontaneously from the tree. Well, he was wrong about that, but Hubbard in 1897 said, I think the fungus is under control of the insect. The insect brings it into the woods somehow. And finally, Helene Franke-Grossman, the single most important scientist in the ambrosia science, said it's difficult to believe they could just accidentally bring this food with them on the exoskeleton. So what she did was dissect cleverly a bunch of different ambrosia beetles and found these big conspicuous organs, and they were full of fungus spores. It was a very clever idea, and it answers a question from earlier. How do they keep their crops? It's with the fungus pockets. It's not just when flying tree to tree, because that absolutely is what they're for. They're full of pure spores. They go to a new tree and they start their new garden, but also over winter without the fungus dying, which is the actual question that drove Helen to seek this answer. Now, she discovered many right at the beginning, but she was calling them general German names like Hauptdrüsen or skin pores, skin glands. But what to call them? Is there a better name? Well, it was the mycologist Raj Batra, who did some of the most important early mid-1900s work on ambrosia fungi, who gave the term Mycanja. And that is the term that I use and the most common term since. Now, if you recognize Lech Batra, that's because he used to be here at Cornell as a student under Korff. Here you can see Batra about a decade before naming the Mycanja. I have found some of Batra's specimens in the teaching herbarium, and it's a pretty cool collection. It's a pretty cool connection. So it was our own Lech Batra who gave it the name while he was working with Lichtwart in Kansas. Helene Franke-Grossman would later say, well, Mycanja is probably more correct, and it kind of is grammatically, but Mycanja has been used much more. Those 5 years of lead time were enough that it took hold, especially in the English-speaking world. Mycanja has been used more in German and other languages and has had a resurgence recently. So birth words are used. Okay, let's get to the most amazing parts of this organ. General features of Mycanja. They are enclosed structures that spores are held inside, narrow openings that the spores come in and exit, and there are gland cells that secrete into the lumen. Inside of the Mycanja are pure fungus masses. They can be actual budding yeasts like in Raffaellia, or they can be phthallic arthric hyphae that break off and disarticulate. In either case, these are dimorphic fungi. They've evolved to turn into this form in the Mycanjum and then go back to a fluffy, beautiful, edible form in wood, and then back again. So all strict mycangial fungi so far have special forms that they take to grow and live in the Mycanjum. There's been great strides in visualization since the early days. I'm not going to linger on this, but it's gone from these beautiful hand illustrations to these beautiful 3D images from combined CT scans. And you can see a few more images here. Here was my best attempt a few years ago at getting this Mycanjum on a microscope, but how can you beat color-coded 3D video game-looking illustrations? It's just incredible. It shows the musculature, it shows the organs in red. It's just extremely cool. And for difficult organs to talk about, it's real. Oh, what's this? But are they just pockets? No, of course not. They're not just pockets. If they were just pockets to hold spores passively, there wouldn't be, I'd be done talking about them. Okay, they hold fungi, that's the end. But they're hardly passive pockets. They are ringed with active gland cells that secrete into the lumen. I already mentioned that, right? These support selective growth of ambrosia fungi. And this is information that had been in the literature for a long time, but kind of under the radar. It had never really been combined together. And it was first tested experimentally in this wonderful paper from 2019, where for the first time, different strains of ambrosia fungi were artificially put into different Mycanjia to see what would happen. And it turns out that Mycanjia aren't just selective in a broad sense, they can actually have quite specific selection on different closely related ambrosia fungi, although there's a mountain of caveats there too. This was my biggest impetus for putting this review and this chapter together and getting this information out, was that this had never been put together in one place, what I'm calling the Mycanjump Cycle. It's really a theory, but it has so much literature to support every step individually, at least in the big Mycanjia, that it is a solid theory and mechanism. So first, the ambrosia beetle has an empty Mycanjum. Propagules come in from the walls. We know that they aren't very careful about this. Molds, nematodes, mites, all kinds of things get into these Mycanjia, and hopefully some of the actual ambrosia fungus, the thing that's all this fluffy white stuff in the gallery. The glands recognize this somehow, and they turn on when the fungus enters, filling the Mycanjum with something. We're actually not sure what. The propagules propagate, but the non-Mycanjum propagules don't. And through simple numbers, through a numbers game, the mutualist, which is tuned to this environment, grows quickly and pushes out fungi that aren't tuned for this environment. Now, this doesn't mean there aren't Mycanjum passengers that have adapted to this. There definitely are. There's great evidence that there's yeasts adapted to live in here. There's great evidence that other fungi also go along with nutritional mutualists. They may also have other mutualistic roles. But in general, this is how molds and other common contaminants are gotten rid of. The beetle flies to a new tree. The fungus continues to grow, overflow from the Mycanjum. I've seen this myself. They're just got a big glob of it coming out, and they just have to touch the wall of their new tunnels to start their new gardens over again. The gland cells turn off at some point. The insides deteriorate and are digested, and we start the whole cycle again. The next generation pick up the fungi when they mature. And this Mycanjum cycle is a really beautiful and important thing, because this is what allows everything. This is the reason ambrosia beetles can keep their special delicate cultivars, despite there being so many other aggressive fungi. This is how ambrosia beetles were able to domesticate their crops in the first place. This is how ambrosia beetles make sure they have pure inoculum when they go to wood. And it's really important to get a good inoculum. For anyone who's ever grown mushrooms at home, you know that for a fact. If you've done oysters or shiitake or organoderma, gotta have a pure inoculum. So, let's talk about the fungi a little bit. And I'm going to go very quickly through this, because this is usually what I talk about, and this was what most of my thesis was about. But Mycanja define fungal cultivar. The Mycanjum that a beetle has defines what kind of ambrosia fungus that beetle farms. The smaller Mycanja generally have Rafflesia ambrosia fungi, and they share them. The same fungus between different beetles. One beetle can have more than one Rafflesia. They are not all that picky. But the bigger fungi, the bigger Mycanja, things like the coiling hosepipe tubes, things like the big membranes inside the body, they're a lot more precise. All of them have ambrosia fungi in a totally different order, the Stratocystidaceae. And I should say, Rafflesia is closely related to Dutch elm disease and Ophiostoma. These Stratocystidaceae ambrosia fungi are closely related to Ceratocystis, a very different tree pathogen with very similar ecology. Each of these Mycanjum types is strictly associated with one genus of the Stratocystidaceae, meaning there's some kind of higher level tuning happening here between insect organ and fungus somehow. And these Mycanja always carry pure masses of ambrosia fungi, except for when they have nematodes or other things in the Mycanja. The Mycanja can actually recognize a nematode inside of them and shut down. And we think that's some of the reason why some ambrosia beetles have doubled Mycanja, so that one can be shut down. I apologize, there's some grass cutting going on outside, and so if there's some background noise, that's why. The prothoracic Mycanjum of this group of beetles only has Phyalophoropsis ambrosia fungi, and it's the same fungus in the Mycanjum that grows right along the walls that the larvae eat. The mesonodal Mycanjum only has Ambrosiella fungi. The prococcyl coil Mycanjum only has Meridithiella fungi. We proved this around the world with multiple species for each of these groups, and erected a new dinner for them. It was part of my thesis, and I really love these fungi. But this answers a question from earlier. What is this mysterious tube? I think you've guessed by now, but it is the spore pocket, the Mycanjum of, in this case, the genus Ortholis. It's my favorite Mycanjum because it's so ridiculous. It's a hose pipe that enters through the armpit that the beetles can squeeze fungi out of. It doesn't get much better than that. The only better one is, well, this one, because it's like a UFO whale blowhole, also toothpaste squeezer. So if you cut these beetles open, the Platypodinae, there's a little UFO in there, and they've got a blowhole on their back, and there's a little onion bulb looking thing that fills with spores that overflow out of the blowhole. And here's a photo of the inside of one. They are filled with these beautiful hairs that guide the spores up to the hole and help them escape. And it's thought the beetle can like move its head back to squeeze on that Mycanjum as well. So these Mycanjum fungus associations have very strong taxonomic patterns, as we showed in Pursunia in 2020. And there's coevolutionary signal there as well. I don't want to get too far into that. I am looking at the clock. I want to talk about one cool story, side story. Xylotrius pallidus, the only ambrosia beetle we know of that has two different Mycanja. That's not new news. Lawrence Abrahamson, under the direction of Dale Norris, worked on these in the late 60s as part of his PhD work, made beautiful slides. So he graciously lent me the slides to take this color photo. There's one of the Mycanja, and there's the other near the head. Two different Mycanja. One is only in the females, one is in both sexes. Again, this is unknown in any other ambrosia beetle. The Prothoracic fungus, which Abrahamson had isolated but never characterized, we were able to isolate and characterize just last year, and we named it after Dr. Abrahamson in honor of his actual first discovery back in the 60s. And the genus is after Anomythiacin Carrick, a very respected forest entomologist who just turned 100 recently. The oral symbiont is another Rafflesia, this really promiscuous common ambrosia fungus group. And so this is interesting. Why does the beetle bother to keep both Mycanja over evolutionary time? Why does it have both fungi? They both grow in the gallery. What's the point? It's a great question. I don't have the answer. So let me go back again to this variety, because I'm not going to cover every one, and just appreciate how many different kinds of Mycanja there are. And it's because every ambrosia beetle clade, I told you earlier that there's 12 different inventions of fungus farming in bark beetles. That's each of these blue clades. Each of them represents this amazing discovery that, hey, I can keep a really good fungus with me, go into the bark, below the bark into the wood, and farm it for food. And each of those seems to have required Mycanja, because not a single one doesn't have some kind of Mycanjum or spore pocket. The hypothetical evolution here is that superficial passengers that get really good at being transferred on beetles, like Dutch elm disease, for example, eventually make their way into some kind of internal structure that lets them be carried even better. And maybe they start to be even better food, so the beetles aren't just passively grazing on them anymore. And then the fungus loses the ability to ever disperse itself. That's what I think of as ambrosia beetles. And the Mycanjum is the lightning bolt that lets that happen. What's also important here is that there's a domino effect. Once there's a symbiont ready for other beetles to farm, it is available for free. Mycanja appear to be prerequisites for every shift from bark beetles to fungus farming. Although again, some bark beetles have Mycanja and do rely on fungi. Now, ambrosia beetles aren't the only animals that farm. We're one of them too. And just like the other animals, domestication requires genetic bottlenecks. Mycanja probably allow that, at least I think so. Every generation, only a few propagules into the organ, propagating selectively to make the next inoculum, perfect recipe for domestication and coevolution. And I talked about that domino effect. Platypodinae, that other family that's all ambrosia beetles, probably domesticated with Rafflesia more than 100 million years ago. And then there were trades that allowed multiple groups to start farming in the Scelidonae, this passing the torch, so to speak. And then some of those groups, for whatever reason, got rid of this and picked up the Ceraticidaceae ambrosia fungi. Wow, that's the first time I've never been able to pronounce that word. It's an important family. We don't know why, but for whatever reason, they seem to need bigger Mycanja for it. And gosh, I could go on and on about all the different ambrosia fungi that have been discovered recently, but that is for another time in the future, because today is about pockets. And those pockets do seem to predate in evolutionary time the fungi that they carry, proving you need a good solid pocket before you get a stable crop lineage. So I'm gonna move towards closing up here because I believe I've got about 6 minutes left. I want to encourage questions. But if each beetle Mycanjum fungus trio is a unique symbiosis, then Mycanja define that symbiosis. And they're super important. It's not just beetle and fungus, it's beetle, fungus, Mycanjum. They've evolved a bunch of times. That makes them a beautiful model for studying the evolution of symbiosis. These should be a wonderful lab system. Where else do you have 12 convergent evolutions to the same organ doing the same thing but in different ways? Imagine the different kinds of tests that can be done on that. But do pockets matter? Of course, I've just spent almost 50, very dangerously close to 50 minutes talking about that, and this beetle's got a backpack on its back. It obviously thinks that they matter, but I can justify them a little bit for a few different reasons. This is why I think it's important to learn about and appreciate Mycanja. I do appreciate the little cartoon beetles keeping me on track. Otherwise, I'll tangent forever. Future proofing, understanding what fungi can actually be vectored by the next Laurel Wilt. What's the next disease to come here? Well, we don't know that unless we know Mycanja. Connecting potential invasive to native susceptible plants, Mycanja kind of feel like the missing puzzle piece there. There's no such thing as overusing an analogy, right? Or a metaphor. Okay. So ecological context. Oh my gosh, the amount of fungi associated with ambrosia beetles is so complex. Some are pathogens, some are nutritional mutualists, some are carried in Mycanja, some are carried elsewhere. Most of the fungi are poorly mapped into this system and exist on a gradient. And it's really only the ones up here that are the true co-evolved nutritional symbionts. But how do we know ecological context? How do we connect an evolving lineage of insects to an evolving lineage of fungi? Oh, I think Mycanja might be the missing piece there. It leads to precision in management. How do we know how to manage problems, whether it's in the orchard or the nursery or elsewhere? Targeting the Mycanjum is a pretty good place, if not targeting the fungus, but how do we know where the beetle microbiome connects to the fungi that might cause disease? I think it's Mycanja. What is in the Mycanjum? What's being transmitted by the beetle? Symbiotic theory. How do ancestral relationships, how did they evolve to become modern mutualisms? This is the study of how all symbiosis evolve, which can extend to parasitisms and mutualism. Mycanja are critical to ever figuring this out in ambrosia beetles, and it is not figured out. Because what we still don't know, there are so many Mycanja that we don't even know the fungi in them. We don't know what the gland cells are actually secreting. We don't know how Mycanja recognize fungi and vice versa. We don't know the triggers for fungi or beetle. And there's a working group called the Bark Beetle Microbiome that I'm a part of, and I've been happy to be a part of, which is working on starting to answer many of those problems and putting out resources to help others do it. So in conclusion, oh gosh, I'm not gonna read all that. Mycanja are very important. And if you agree, look out for this book chapter that's coming out soon that goes over all of this in detail and argues the point that Mycanja are essential. So with that, I want to thank all my collaborators. I'm sure I'm forgetting somebody here that without them, I couldn't have made the few studies I mentioned possible, connecting Mycanja to fungi. Of course, my PI from, gosh, feels like 2 lives ago back in the PhD, who was wonderful and helped all this work. I also want to thank Teresa Polaska and the whole Polaska lab here for everything. More about that next time you see me. But thank you so much for listening to me talk about pockets. I will welcome any questions. Thank you so much.
Thank you, Chase. This was beautiful. Thank you. Are there any questions for Chase?
Might not be time.
No. We'll take all questions. Questions.
I have too many questions. Great topic. Is there a way of studying the yeast to filamentous fungal transition? Would that be a clue?
Yes, there is. In fact, like most things in ambrosia beetles, and I didn't get to pontificate about how important it is that each of these is a separate evolved group, and way too often people make assumptions across them on beetle and fungus. We have a pretty good clue about how this happens in the Ophiostoma thales, because Ophiostoma novo-ulmi and Sporothrix and other Ophiostoma thales have density-dependent quorum-like mechanisms to go from between states. And we've gotten, I should say, Tom Harrington had preliminary data showing the same in Rothielia, not enough to prove, but it seems like a good hunch. In the Stratocystideceae, we have no idea. I can say it didn't seem to me like density-dependent stuff worked, but yes, it is a way to do it.
I have a follow-up question related to Gillian's question. Can you actually obtain yeast that you find in the pockets in vitro?
Yeah, can you explain what you mean by yeast? Do you mean the yeast form of the
so you mentioned that some of the pockets are populated by yeast and others by spores that are kind of arthrospores, but not hyphae.
Yes, yeah, you can in vitro. So can you make basically a test tube mycanjum? Is that what you're asking? It hasn't been tried to my knowledge, but it would be cool to do. And I think it's one of the obvious steps in figuring out how the mycanjum works. Would be to try to replicate the compounds. We know from the 60s the compounds are oily, greasy, very lipidaceous, but also very full of amino acids. But it's not clear what exactly they do. It's not clear if they actually feed the fungi through selective food, or if they just make a conducive environment that the fungus can handle better. For example, maybe they handle lipids better because, okay, I'm going out there, but maybe because they evolved from fungi good to sticking to lipidaceous beetle surfaces. It's a logical ancestral history to me. But yes, mycanjum in a test tube would be wonderful.
Thank you. There is a question from Chris in chat.
Yes. Yeah, is it known how the primary fungus at the beetle farms removes spores from other fungi or bacteria? And so I'm not exactly sure what you mean by removes spores, but I think you mean in the Mycanjump cycle when other spores are excluded from the Mycanjum.
Exactly. How does that exclusion happen?
Just through competitive exclusion. And again, this has not been demonstrated. This has not been demonstrated in action in the laboratory, which I should give as a caveat. But we know that before the glands activate, the Mycanjum can be really dirty. They can have all other molds. They can have dirt. They can have brass, wood, and a couple of ambrosia spores. And after the Mycanjum glands activate, they fill with almost entirely just the mutualist. And we know that when they're in flight, by that time, it is like 99.9999% pure mutualist. And the mechanism that makes sense is that the other trash and gunk in there was pushed out. And there's actually been beautifully illustrated cross sections of Mycanja over the year showing, and it's a lot of junk, going to pure, like beautiful mutualist spores.
Wow.
You mentioned defenses, 3 defenses, and I kind of missed this bit. So beetles obviously would induce defenses, but they are different defenses than fungi would induce, because beetle defenses are the fungi defenses. 3 defenses against insects are jasmonic acid based, and against fungi are salicylic acid based, in kind of generalizing. Can you talk about this a little bit, or elaborate, elucidate?
I can, and those defenses vary a lot. I'm not an expert in them, but they vary from really heavy resin production to other active defenses in the tree that make things bad for the beetles. But because the beetle is the vector for the fungus, and the fungus cannot get in without the beetle successfully digging in, most ambrosia beetles don't have the behavior in the first place to attack living trees. They actually look for ethanol that's given off by the process of decomposition. That is their major attractant. So behavior is the biggest barrier to ambrosia beetles going into living wood in the first place, meaning the fungus can never go there to meet these defenses. But second, an ambrosia beetle that digs in and realizes it's made a mistake will usually leave, and it will do this before it's in the sapwood or before it's had a chance to establish the fungus well. There's more competitive fungi in the phloem, or maybe they just don't get in deep enough. But when this breaks down is when disaster happens, because The red bay ambrosia beetle, the one that causes laurel wilt in the US, has an unusual biology, one that it also does in its native range in China of having the behavior to attack living trees and to dig in a good bit. Now, it backs out again afterwards and tries again, but it digs in enough to inoculate the fungus. And once the fungus is there, it does indeed overcome the tree defenses and cause disease. And so this is kind of how laurel wilt is first introduced. It's once the tree declines more that the beetles can come back and really colonize. So yeah, I would say absolutely the defenses are different. It's the beetle behavior that makes the difference. And they are very particular.
Thank you. Any other questions?
I'll venture a question.
Yeah, please.
Yeah, so I was just curious if there's any entomopathogenic fungi that prey on these beetles, or if any generalist insect pathogens can infect these beetles, and whether there's some degree of recognition of their fungal partner that distinguishes between mounting an immune defense, perhaps. I don't know much about insect immunology, though, I imagine.
That is an excellent, that is an excellent question. Because entomopathogens and fungus pathogens have been a really big deal in other fungus farming systems. Now, I can answer with the very general, yes, there are entomopathogens that kill ambrosia beetles, and they have indeed been introduced as control mechanisms for ambrosia beetles. Now, I'm going to put my foot in my mouth here, but I think it was Bavaria bassiana and one more. I could do a down low Google search, but there's no honor in that. So there have been at least two entomopathogens that have been used for insect control. I'm not the one to answer that. The one who is Luella Castrillo at USDA ARS here in Ithaca, as well as John Vandenberg, who is there, who have both done work on exactly this in the field, and I'm sure many others. So I'm not the expert to answer that, but yes.
Okay, cool. Yeah, I worked a little bit on Bavaria. I know it's a super generalist, so that would make sense.
Yeah, and it turns them pink. It turns the larvae pink.
Yeah, it does.
Which is fun, you know, you get a little fun with your control.
Yeah.
Yeah,
thank you.
Good question.
Hi, Chase. I do have a question.
Yeah,
I
sure.
find it really stunning that ambrosia fungi can thrive in really oily environments of the Mycanja of the insects, and also these really cellulose and lignin-rich environments of the tree. Do you think that has any connection to the dimorphic lifestyle of these fungi? Has anyone ever tried growing the hypho stage in like these oily rich environments, or vice versa?
Sorry, I didn't quite catch the end. Growing in a what rich environment?
Hi. Has anyone ever tried growing the like hypho stage of these ambrosia fungi in these oily rich environments, and the sort of yeast-like stages in the like cellulose rich environment?
Was it protein rich environment? Did you say protein rich?
Oh, if it was protein rich, yeah. Sorry, I think I must have misheard lipid rich.
Oh no, lipid rich environment. Yes, yes, yes. Gosh, it's been a little bit, but following the advice of the expert, Helene Franke-Grossman, the most important ambrosia fungus researcher, who said you could pour olive oil on the cultures and get them to change. I tried it. I didn't get it to work. You know how that works. Sometimes it takes a magic touch. I did try growing them on sterilized wood chips. I've tried proline additions, which is supposed to be an amino acid that will help trigger mycanjal growth. Others, you know, folks decades ago said they got it to work. It was one of those little side projects that never yielded, but certainly it has to be possible. They also respond to beetle grazing. So just the act of a beetle eating the fungus can be what it requires to start making food, to actually fruit. Otherwise, they'll be sterile. And so some authors decades ago would let little beetles walk around in their cultures and eat, and it would cause sporulation, because there's really 3 forms of the fungus, something I didn't get into. The mycanjal form, where it can divide and grow and you know, proliferate. The filamentous form, where it grows into the wood and extends. It doesn't do any wood decay. These are not wood decay fungi, but extend and extract nutrients. And there is the ambrosial form, where it commits all of its energy to like completely ridiculous spores that are really tasty. All 3 of those, it switches between under different triggers. But yeah, I have tried similar culture manipulations, just no luck.
A really fascinating system.
Absolutely. Yeah, there's just so many avenues just begging to be explored under this symbiosis. And then you can do it 12 times, because once you do it for one group, you don't know it evolved the same way. So yeah, thanks for that question.
Excellent. Other questions?
Hi, Chase. Great talk again. One question, just follow up to Tristan's point, or just follow up to your answer. You said they will change basically their sort of like life stage, whether they proliferate or not. When they switch this environment, have anyone tried to sort of like trigger that in laboratory conditions?
The closest to that, well, sorry, triggering what part? The fungus responding or the Mycanjum responding?
The fungus or the Ambrosia fungus to trigger that into sporulation, like sort of imitating when it's changing from Mycanja to, for example, the wood. Yeah.
Yes, it has been tried. A couple caveats. So one, the reverse was tried back in the 70s by Isolde Schneider, who was a legend of metabolism and glands, and was the single most important study in my Mycanjump Cycle theory, because she actually looked at the Mycanja and watched the glands activate when the fungus touched them, and then grow only instead of the other fungi in the secretions, and so did observe that direction, the filamentous to Mycanjum form. You don't have to work hard for the other direction, because plating the Mycanjal form on media, it just goes to filamentous. It's not that it needs a trigger to go filamentous. It seems to be that it loses the context, the microenvironment of the Mycanjum, and then goes to filamentous as its default state. So it's really just a binary. Is it in the Mycanjum environment or not? If it is, turn into a Mycanjal state. That could be enough for the advantage they have in the Mycanja, is recognizing that they're in one and starting to grow and proliferate as a single cell, when the other interlopers, and I don't like to use that term because it's loaded, the other fungi might have roles in the Mycanjum, but maybe the other interlopers don't know that. They're wasting time as hyphae and they get pushed out. Just a theory. But yeah, it's observed all the time. It's the best way to isolate them, the fungi from the beetles.
Any other questions? I don't hear any. Chase, thank you very much. This was wonderful. Thank you. And good luck with your Ambrosia fungal pursuits. And thank you very much, guys.
Thanks for having me.
We'll see you next week, I guess.
Thanks, Chase. Thanks.
Thank you.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.